Our next speaker is Dr. Oren Kraus. He is the director of machine learning at Recursion Toronto. His group focuses on advancing machine learning for tech-enabled drug discovery. And today he will be talking about foundation models in early stage drug discovery. So with that, we'll let, so Oren, he cannot come in person, so he will be sharing his talk on Zoom.
Hey everyone, really sorry I couldn't be in person at MLB today. I was at the airport most of the afternoon yesterday, but because of the flight disruptions in Boston Logan, I wasn't able to come in. First, I want to say I'm a huge fan of the symposiums at the Broad Institute. Back in 2016, I met my co-founder of my previous startup at a Cytodata Symposium that Ann Carpenter's lab organized. So it's really a great opportunity for everyone there to take advantage of it and network. So my talk today will be about foundation models in early drug discovery. So I'll introduce Recursion's platform first and what we call maps of biology. And I'll specifically talk about how we build foundation models to analyze the high content data that we generate at Recursion. And finally, I'll plug that into how we can use those foundation models for virtual screening. So Recursion started about 12 years ago. The initial idea was focused around using deep learning that was really taking off for computer vision at the time for microscopy data, and the idea that we could really extract very high-level insights from phenotypic-based images. We've expanded a lot since then, so much more than phenomics going on at Recursion. We have high-throughput transcriptomic assays, also a lot of work going on in computational chemistry and just general ML/AI research. And currently we have 4 clinical stage programs, 3 in oncology and 1 in rare disease, and partnerships with leading tech companies like NVIDIA and Google, as well as leading biopharma companies like Sanofi and Roche Genentech. So the core concept that Recursion has is to change the shape of the drug discovery funnel. So we actually want to expand the scope of therapeutic areas that we're exploring and potential targets that we're exploring in those therapeutic areas with an unbiased genome-wide screen up front. And then we want to use predictive models to quickly assess which of those are not really feasible so we can fail them fast. And finally, we're using cutting-edge computational chemistry tools to accelerate how quickly we can develop compounds against those potential targets. This is powered by our robotic wet lab, which can conduct 2.2 million wet lab experiments every week across approximately 50 different cell lines. And we use whole genome CRISPR for most of these maps that we built. We've screened over 2 million compounds, totaling over 65 petabytes of proprietary data, mostly imaging-based. So how can we use phenotypic models for drug discovery? So I'll start with a straightforward approach. So in this case, we have a clear disease model and healthy controls. So when we run imaging experiments with these, we can look for compounds that revert the disease model back into the healthy phenotype. And to do that, we need really good representations of what's going on in the images, ideally ones that can deal with all the batch effects that show up between different experiments. And that's where I'll spend the bulk of the talk today, is how we generate those. But once we have those embeddings of images, we can start to compare different conditions. So in this example, we're looking at a disease model of SARS-CoV-2. And we can generate a vector in high dimensional space that indicates the direction of the phenotype from the control condition to the disease condition. And then as we start screening compounds, we can try to see which ones revert the phenotype in the direction of the disease. So we can project those vectors onto the healthy disease vector and ideally find compounds that have a strong on-target effect. So for COVID specifically, the team at Recursion went through a mega effort back when COVID was just starting to contribute to the scientific community. So we spent about 4 weeks screening all the potential treatments in a variety of cell lines. 2 well-known treatments that we screened were remdesivir and hydroxychloroquine. So in these examples, you can see how different compounds have on-disease and off-disease scores. So remdesivir and this GS compound have positive responses with increasing doses, but the drugs like remdesivir seem to have very high off-target effects. And when we expand this to all the compounds that have been considered in clinical trials, it took a few years to have all the readouts for these, but the platform essentially predicted 9 out of the 10 clinical trials correctly in terms of efficacy. So it really shows the power of doing phenotypic screening for drug discovery. So moving on, I'll explain another interesting application of phenotypic screens, especially when we start adding genome-wide CRISPR knockout screens. So in this case, we can start to recover known functional relationships in genetic networks. In this example, we're looking at the mTOR pathway. And if we look at the same phenomic vectors in one of these knockout screens, we can see that genes that are part of the same complex will typically have high similarity. So in this case, TSC1 and TSC2 seem to have like similar phenomic embeddings. And things that are inhibitory of that complex have an opposite effect. So we call these phenol opposites typically. So this lets us recover known interactions, but we can also start to uncover novel interactions. So in this case, if we're screening compounds in the same space, we can also find compounds that have a similar effect to a gene knockout or an opposite effect to a gene knockout. One of the applications of this is we can start to go after targets that are hard to develop compounds against in traditional settings. So one example is MYC, which is disordered, so it's hard to do like a typical binding assay against it. But when we look at in the phenotypic space, we can identify a handful of compounds that seem to have dose-response relationships with MYC. This doesn't necessarily mean that these compounds are directly interacting with MYC, but they seem to have a similar phenotypic effect. And then when we do this across the whole proteome, more or less, we can start seeing proteins with similar annotations clustering together. And similarly for compounds with known annotations, they cluster together in these phenotypic spaces. And if we expand to genome-wide screens, that's really where the magic happens, where we can start to identify completely novel interactions between genes and genes or genes and compounds that weren't previously known. So that enables us to really open up the hypothesis space for new targets in therapeutic areas. To benchmark these, what we call maps, we developed a framework along with collaborators at Genentech. So this can be applied to cell-painting images or even RNA-seq data. But we start with the embeddings I was mentioning. First, we filter out any QC issues from those samples. Then we use batch alignment techniques to align all the experiments so we can get good representations. And finally, we aggregate those to the perturbation level. And that's when we can start to relate perturbations to each other. For benchmarking, there's 2 main categories of benchmarks we have. One is to identify how many perturbations have a signal that's differentiated from the background effect. And the other one is looking at interactions between perturbations. So here we rank all the similarities and different opposites between all the perturbations. And we're looking to see in the extremities of that distribution how many known interactions are recovered based on databases like StringDB and Corem. So with that background, I'll explain how we train these embedding models to extract information from the images. Our initial approaches, which were the first foray into deep learning after we moved away from Cell Profiler, was using what we call weakly supervised learning. So this approach, we take image crops from the microscopy wells, and then we try to predict what the perturbation was that was applied to those wells. This was done with a data set that we released publicly called RxRx1. This was part of a NeurIPS challenge in 2019 for the computer vision community. So that data set has about 1,000 sRNA treatments across 50 batches. So it's a good way to get the model to learn how different perturbations look versus the batch effects, which are kind of labeled the same way over and over again. Once this model is trained, we can use the second to last layer typically as the feature representation. And that's what was used to power the maps in our platform for a while. One issue we faced with this as our data set grew is that when we wanted to train larger models with more diverse data, we found that performance typically degraded. And that's what motivated us to start looking at self-supervised learning approaches. So these were becoming very popular around 2022. So in this setting, instead of using the labels of what was applied to the wells in the microscopy data, we're just using the raw microscopy images as the training data. And the task here is to reconstruct masked patches in the original image. So typically this is done with a high mask ratio of around 75%. And then our transformer-based encoder only really sees the visible patches, and it's tasked with reconstructing all the pixel values from the masked patches. The nice thing about this is that it can scale really well, so we can train much larger models and much larger data sets, and that tended to increase performance, which I'll show in the next few slides. And the embedding we use here is like the output of the transformer encoder for the decoder layers. So just for some stats on this approach, we were able to switch from a DenseNet backbone to a vision transformer backbone. That allowed us to increase the model size from 30 million parameters to 300 million, so 10x improvement. And then, the biggest challenge was the dataset size. So instead of using just those 100,000 wells that we had for RxRx1, we expanded to nearly like all the phenomic wells that Recursion had that we were able to train this on. So that was 1,000x increase in the dataset size. And obviously that took a lot more compute. And then this work was presented at CPPR last year. So overall, the resulting model was a VIT large with a patch size of 8, 330 million parameters, trained on those 93 million wells for 40 epochs. And it really showed that, the same recipe that's being applied in the general NLP and computer vision communities also applies to these biological data sets, where if we scale compute and scale the model size and the dataset size, we're seeing improved performance, specifically on that recall benchmark I was mentioning. So what we did next, so this happened last summer. Recursion upgraded our cluster, and for a while it was number, I think, 35 or 36 of the top supercomputers in the world. And this work was a bit driven by Iman Haq, who's in the audience, but we wanted to push this even further. So we expanded the model size to a VIT gigantic, which has 1.8 billion parameters. That's a 6x improvement over the Phenom-1 model. And then it was trained for 4 times longer on the new cluster that we installed. And interestingly, it actually was trained on a smaller data set. So we took a subset of the 93 million data set, and I'll explain why in the next few slides. So this work was presented at ICML this year. What we showed pretty quickly was that with this approach, we continue to see improved performance with scale. So here you're looking at a log plot of the model parameters against the relationship recall in StringDB. So that 1.8 billion parameter model is once again improving over Phenom-1, almost linear in the log space. And additionally, on the replicate consistency metric, it also had a pretty significant improvement. So about 25% more knockouts were found to be consistent with this model. In addition to just scaling, we actually had to make quite a few changes to this approach to get it to work better. One is what I mentioned about data creation. So the original 93 million data set had mostly like everything we could scrape from Recursion. But what we did here was try to reduce redundancy in the data set, so it was more diverse and could provide stronger signal over the training batches. So we removed a lot of negative controls, which tend to look similar to each other, and prioritized wells that had consistent signal using other models like the weakly supervised model or the Phenom-1 model. And the other key insight we had is the final layer of the encoder doesn't necessarily have the best feature space for the downstream tasks. So we came up with some methods to find which layer from the encoder actually had the best performance downstream. So the plot here on the right shows how each one of those contributions contributed to the performance across both metrics. Diving deeper into that blockwise search I was mentioning, it's really impractical to run the recall metrics across every single one of the 48 layers for the largest model across all the checkpoints. because we need to inference 80 million images each time because we have to compute the embeddings for the entire genome knockout. So what we came up with was 2 proxy tasks that are based on linear classifiers at intermediate layers. So one of these is a set of 40 genetic functional groups. So just a 40-way classification. And the other one is the RxRx1 dataset I mentioned previously. So these are much smaller datasets to work with. And we can see that the performance kind of degrades toward the later layers, especially for the larger models. So that let us select like the ideal layer for downstream tasks. And then we showed that the proxy tasks are actually well correlated with the more compute-intensive recall and replicate consistency tasks. So these have a rank correlation of 97 and 91% for the string DB recall and the replicate consistency recall, respectively. So overall, the best model was what's called an MAE gigantic model with a batch size of 8, and the trim model, which only computes up to the 38th layer instead of the 48th layer, had the best performance on the replicate consistency metric and on the recall metric. And you can see the effect of individual changes. So the data set creation has a big impact on the replicate consistency. The layer selection does as well, and same with the scaling. And then finally, we also evaluated this model on external data sets. So we computed the same recall metric on the JumpCP data set. This is a different set of gene knockouts, so it's not directly comparable to the RxRx3 experiments. But there we also saw that with the larger model sizes, like the recall generally improves even on these external data sets that actually have a different set of channels on them. So that was quite impressive. To support the scientific community, we've actually made these models and some of the data sets publicly available. So OpenPhenom is a smaller version of the Phenom models I was mentioning. It's actually trained with a channel agnostic architecture. So whether you have one channel or I think up to 12 channels, you can still compute embeddings from it. And then we released it completely open source on Hugging Face, and it's been downloaded over 115,000 times since about last year. RxRx3, that's a genome-wide knockout screen that also includes about 1,600 compounds. So it's much larger. It's about 83 terabytes, but it's also been downloaded 6,000 times. And then RxRx3 Core is a subset of that data set that's on Hugging Face. And it's more designed for benchmarking, and it has an associated benchmarking task. So that one was published at a workshop at iClear last year. I only have 1 minute left, so I might have to skip over the other application about virtual drug screening. But this was work that was done at Valence, where we combined graph encoder models, specifically one called GPS, with these Phenom-1 embeddings to be able to train contrastive models that can virtually screen compounds against phenotypic profiles. So the takeaway there is that the foundation model for phenomics is a pretty key component in getting contrastive models to work with graph neural network-based approaches on molecules. So yeah, with that, I'll wrap up. The key takeaways are that self-supervised learning combined with data creation and scaling and compute and data set sizes is a really good recipe for training foundation models for phenomic images, but also generally for scientific data. And that as we start to combine foundation models, it really unlocks new capabilities for multimodal learning. And with that, I'll take any questions, and thanks for listening.